Block of polyspermy. Fertilization is a prime event in the development of an embryo. Embryonic growth and development starts with the fertilization of an ovum by a spermatozoon to form a unicellular zygote, which eventually divides and grows into an embryo. Hence, the sperm is first recognized by the oocyte vitelline membrane or by the cells surrounding the ova, known as zona pellucida. This is followed by scattering of the zona cells and the lysis at that region of the ova envelope by the release of sperm acrosomal enzymes. This lysis is actually followed by the fusion of the sperm plasma membrane with the plasma membrane of the ovum. The entry of a sperm into the ovum involves a binding process facilitated by the formation of an extension. The entry of a sperm into the ovum involves a binding process facilitated by the formation of an extension of several microvilli. The entry of a sperm into the ovum involves a binding process facilitated by the formation of an extension of several microvilli to form a transitory fertilization cone, which appears to be extended by the polymerization of actin. Once again, I'm going to read this. The sperm first recognized by the oocyte vitelline membrane or by the cells surrounding the ova, known as zona pellucida. The sperm is first recognized by the oocyte vitelline membrane or by the cells surrounding the ova, known as zona pellucida. This is followed by scattering of the zona cells and the lysis at that region of the ova envelope by the release of sperm acrosomal enzymes. We know that. This lysis is followed by the fusion of the sperm plasma membrane with the plasma membrane of the ovum we mentioned. The entry of a sperm into the ovum involves a binding process facilitated by the formation of an extension of several microvilli to form a transitory fertilization cone. The entry of a sperm into the ovum involves a binding process facilitated by the formation of an extension of several microvilli to form a transitory fertilization cone, which appears to be extended by the polymerization of actin. The entry of sperm into the ovum involves a binding process facilitated by the formation of an extension of several microvilli to form a transitory fertilization cone that appears to be extended by the polymerization of actin. The sperm and ovum plasma membranes then fuse. The sperm head and nucleus pass through the releasing cytoplasmic bridge. This process of fertilization is similar in most species during the fusion of gametes. The protein fertilin then helps to bind the sperm plasma membrane to the ovum membrane, fusing them together. When the membranes are fused, so the process of fertilization is similar in most species during the fusion of gametes. The protein fertilin then helps to bind the sperm plasma membrane to the ovum membrane, fusing them together. When the membranes are fused, the sperm nucleus, mitochondria and centriole can enter the ovum. This event marks the culmination of fertilization. However, nature has designed specific means to avoid the error of allowing more than one sperm cell to enter the ovum being fertilized. This phenomenon is termed as block to polyspermy. The blocked polyspermy. As soon as one sperm has entered the ovum, the fusogenic nature of the ovum membranes, which was mainly to enable the entry of sperm inside the ovum. As soon as one sperm has entered the ovum, the fusogenic nature of ovum membrane, which was mainly to enable the entry of the sperm inside the ovum, becomes a dangerous property if not changed quickly. Certain mechanisms are therefore initiated to reduce this fusibility of the ovum membrane to restrict the entry of additional sperms into the ooplasm. In most animals studied, any sperm that enters the ovum can provide a haploid nucleus and a centriole to the ovum. Hence, only one sperm should fertilize the ovum to avoid propagation of abnormalities. In normal conditions, monospermy occurs in which only one sperm enters the ovum. A haploid sperm nucleus can combine with the haploid ovum nucleus to form the diploid pronucleus in the zygote. Thus, under normal circumstances, the chromosome number of zygote is restored to the original somatic cell to a number as appropriate for the species. The centriole, which is provided by the sperm, will divide to form the two poles of the mitotic spindle during cleavage, and the process of development is initiated. The term polyspermy is used to describe an ovum that has been fertilized by more than one sperm. Diploid organisms normally contain two copies of each chromosome, one from each gamete, the ovum or the sperm. The cell resulting from polyspermy, on the other hand, contains three or more copies of each chromosome, one from the ovum and one each from multiple sperm. The result is consequently a non-viable zygote. This may occur because sperm prove to be efficient at reaching and fertilizing ova due to the selective pressures of sperm competition. Such a situation is therefore deleterious to the newly formed zygote. The entry of more than one sperm, polyspermy, could lead to serious abnormal consequences in most organisms. For example, in sea urchin, fertilization with two sperm cells results in a triploid nucleus in which each chromosome would appear 3 times rather than twice. Moreover, since each sperm centriole divides to form two poles of a mitotic apparatus, instead of a bipolar mitotic spindle separating the zygote chromosome into two cells, the triploid chromosomes would have been divided into 4 cells. In addition, there is no mechanism to ensure that each of the 4 cells receives the proper number and type of chromosomes. So when we talk about the case of sea urchin, fertilization with two sperm result in a triploid nucleus in which each chromosome would appear 3 times rather than twice. Moreover, since each sperm centriole divide to form two poles of a mitotic apparatus, instead of a bipolar mitotic spindle separating the zygote chromosomes into 2 cells, the triploid chromosomes would have to be divided into 4 cells. In addition, there is no mechanism to ensure that each of the 4 cells receives the proper number and type of chromosomes. The chromosomes would be divided unequally. Some cells could receive extra copies of certain chromosomes and other cells would lack them. As early as 1902, the scientist Theodore Boveri demonstrated that some such cell either die or develop with some abnormality. The first problem that an ovum and a sperm of any organism face after fertilization is the possibility of polyspermy, which would impede successful development of an embryo. So, in other words, polyspermy is not a natural process and hence has to be blocked. Polyspermy is the fertilization of an ovum by multiple spermatozoa, and the results of such unions are lethal for the embryo. If more than one sperm fertilize an ovum, the embryo inherits multiple paternal genomes and many centrioles. The organization of chromosomes for the first cleavage division would be chaotic, and the association of chromosomes to the spindle apparatus would be anomalous. This could cause competition for extra chromosomes and result in the disruption of the creation of the cleavage furrow, thus causing the arrest of zygote development. Hence, the process of polyspermy should be prevented, and definite mechanisms have been studied in various organisms to avoid the ovum being fertilized by supernumerary sperm. This phenomenon is known as block. So, mechanisms, there are some sort of mechanisms with which this polyspermy can be blocked. So, in sea urchin is an important model organism in the study of fertilization and embryonic development, and therefore polyspermy in sea urchins has been studied in detail. The methods of polyspermy prevention used by the sea urchin have been described in 2 main pathways. The sea urchin ovum thus has 2 mechanisms to avoid polyspermy: a fast reaction accomplished by an electric change in the ovum plasma membrane, and a slower reaction caused by the exocytosis of the cortical granules. Fast reaction accomplished by the electric change in the ovum plasma membrane, and a slower reaction caused by the exocytosis of the cortical granules. These 2 primary pathways are known as fast block and slow block to polyspermy. The fast block to polyspermy is achieved by changing the electric potential of the ovum plasma membrane. The fast block to polyspermy. The fast block to polyspermy is achieved by changing the electric potential of the ovum plasma membrane. This membrane provides a selective barrier between the ovum cytoplasm and the outside environment. This membrane provides a selective barrier between the ovum cytoplasm and the outside environment. And the ionic concentration of the ovum differs greatly from the first block. This membrane provides a selective barrier, changing the electric potential of the ovum plasma membrane. So this is something which is happening in case of ovum. Don't get confused. This membrane provides a selective barrier between the ovum cytoplasm and which membrane? Plasma membrane, ovum plasma membrane. This ovum plasma membrane provides a selective barrier between the ovum cytoplasm and the outside environment, and the ionic concentration of the ovum differs greatly from that of its surroundings. This concentration difference is especially significant for sodium and potassium ions. Seawater has a particularly high sodium ion concentration, whereas the ovum cytoplasm contains relatively little sodium. The reverse is the case with potassium ions. This condition is maintained by the plasma membrane, which firstly inhibits the entry of sodium ions into the oocyte and prevents potassium ions from leaking out into the environment. If we insert an electrode into an ovum and place a second electrode outside it, we can measure the constant difference in charge across the ovum plasma membrane. This resting membrane potential is generally about 70 MV, usually expressed as minus 70 MV because the inside of the cell is negatively charged with respect to the exterior. Within 1 to 2 seconds after the binding of the first sperm, the membrane potential shifts to a positive level. So, we were mentioning about the potential difference. Reverse is the case with potassium ion. Okay, so seawater is particularly high in sodium ion concentration. So, seawater is particularly low in potassium ion concentration. And ovum cytoplasm contains relatively little sodium and a great amount of potassium. Okay, this condition is maintained by the plasma membrane, which safely inhibits the entry of sodium ions into the oocyte and prevents potassium ions from leaking out into the environment. If we insert an electrode into an ovum and place a second electrode outside it, we can measure the constant difference in charge across the ovum plasma membrane. This resting membrane potential is generally about 70 MV, usually expressed as minus 70 MV because the inside of the cell is negatively charged with respect to the exterior. Within 1 to 3 seconds after the binding of the first sperm, the membrane potential shifts to a positive level, about plus 20 MV. This change is caused by a small influx of sodium ions into the ovum. Although sperm can fuse with membranes having a resting potential of minus MV, they cannot fuse with the membranes having a positive resting potential. So no more sperm can fuse to the ovum. So minus 70 MV to plus 20 MV after fertilization. So that is caused by a small influx of sodium ions into the ovum. Although sperm can fuse with membranes having a resting potential of minus 70 MV, that means unfertilized condition, they cannot fuse with membranes having a positive resting potential. So no more sperm can fuse to the ovum. Polyspermy can be induced if sea urchin ova are artificially supplied with an electric current that keeps their membrane potential negative. So minus 70, 60 or whatever it may be will be there. So polyspermy will be happening, which definitely going to lead to some abnormalities. Fertilization can be prevented entirely by artificially keeping the membrane potential of ova positive, like tricking, which can be used as a contraceptive method, I guess. The fast block to polyspermy can also be prevented by lowering the concentration of sodium ions in the water. If the supply of sodium ions is not sufficient to cause the positive shift in membrane potential, polyspermy occurs. It is not clear that the change in membrane potential acts on the sperm to block secondary fertilization. Mostly often the sperm carry a voltage sensitive component, possibly a positively charged fusogenic protein, and the insertion of this component into the ovum plasma membrane could be regulated by the electric charge across the membrane. To explain this mechanism, after the sperm receptors came into contact with the outer investments of ovum, lytic enzymes are released and break down the outer protective layer. The sperm head then comes into contact with the vitelline and plasma membrane of the ovum. When the two plasma membranes come in contact, molecular signals in the ovum are initiated. These signals allow entry of the first sperm to bind to the ovum membranes and later block entry of any more spermatozoa into the ooplasm. First, Na plus channels in the ovum open, allowing Na plus to enter into the ovum. This causes a depolarization of the ovum from its normal resting potential of minus 70 MV. While depolarization takes place, the remnants of the barrier layers around the ovum are disappearing. With the resolution of three layers and these layers and the depolarization of the plasma membrane, other spermatozoa do not receive the trigger to undergo the acrosome reaction and cannot penetrate the ovum membrane. Thus, this forms the first means of prevent fertilization by multiple sperm. After the sperm receptors came into contact with the outer investments of ovum, lytic enzymes are released and break down the outer protective layer. The sperm head then comes into contact with the vitelline and plasma membrane of the ovum. When the two plasma membranes come in contact, molecular signals in the ovum are initiated. These signals allow entry of the first sperm to bind to the ovum membranes and later block entry of any more spermatozoa into the ooplasm. First, Na plus channels in the ovum open, allowing Na plus to enter into the ovum. This causes a depolarization of the ovum from its normal resting potential of minus 70 MV. While depolarization takes place, the remnants of the barrier layers around the ovum are disappearing. With the dissolution of these layers and the depolarization of plasma membrane, other spermatozoa do not receive the trigger to undergo the acrosome reaction and cannot penetrate the ovum membranes. Thus, this forms the first means to prevent fertilization by multiple sperm. These two simple changes are part of the first block to polyspermy, and I am quite confused about this second method. So once again, I'm going to read this. The sperm receptor come into contact with the outer investments of ovum, lytic enzymes are released and break down the outer protective layer. The sperm head then comes into contact with the vitelline and plasma membranes of the ovum. When the two plasma membranes come in contact, molecular signals in the ovum are initiated. These signals allow entry to the first sperm to bind to the ovum membranes and later block entry of any more spermatozoa into the ooplasm. First, Na plus channels in the ovum open, allowing Na plus to enter into the ovum. This causes a depolarization of ovum from the normal resting potential of 70 MV, minus 70 MV. While depolarization will be there, so the remnants of the barrier layers around the ovum are disappearing. With the dissolution of these layers and the depolarization of the plasma membrane, other spermatozoa do not receive the trigger to undergo the acrosome reaction and cannot penetrate the ovum membrane. Thus, this forms the first means to prevent fertilization by multiple sperm. These two simple changes are part of the first block to polyspermy known as fast block. It is so termed because the fast block to polyspermy is initiated within 1/10 of a second of gamete membrane contact. Hence, the prevention of polyspermy in sea urchins depends on a change in the electrical charge across the surface of the ovum, which is caused by the fusion of the first sperm with the ovum. So, unfertilized sea urchin ova have a negative charge inside, but the charge becomes positive upon fertilization. When sea urchin sperm encounter an ovum with a positive charge, sperm ovum fusion is blocked. Thus, after the first sperm contact the ovum and causes the change, subsequent sperms are prevented from fusing. This electrical polyspermy block is thought to result because a positively charged molecule in the sperm surface membrane is repelled by the positive charge at the ovum surface. Positive like that. So with such a mechanism of an electric block to polyspermy also occur in frogs but has not been demonstrated in most mammals. The fast block to polyspermy. Next method. Fast block to polyspermy is transient since the membrane potential of the sea urchin ovum remains positive for only about a minute. This brief potential shift is not sufficient to prevent polyspermy, which can still occur if the sperm bound to the vitelline envelope are not completely removed. So this process takes place the cortical granule reaction, a slower mechanical block to polyspermy that becomes active about a minute after the first successful sperm ovum attachment. While the first fast block is quite a simple process which takes place very fast. The second block to polyspermy, the slow block, takes place 10 to 60 seconds after the initial penetration of the ovum's plasma membrane by the sperm. Take place 10 to 60 seconds after the initial penetration of the ovum's plasma membrane by the sperm. In this pathway, several molecular and physical changes take place in the sperm gamete membranes. Once the sperm has made its way through the outer layers, receptors in the vitelline layer pass on a signal confirming the entry of a specific sperm. In organisms with external fertilization, such as in sea urchin species, this step confirms that the ovum has been correctly fertilized and it is not by sperm from any other organism. So once sperm binds to recognition molecules or receptors, is confirmed to be compatible. The plasma membranes of the sperm and ovum then fuse. Fusion of the two membranes cause a release of inositol triphosphate. Fusion of these two membranes cause a release of inositol triphosphate. So there will be inositol triphosphate or IP3 release. Triggers release of Ca2 plus into the ovum cytoplasm. So the fusion of the two membranes cause a release of inositol triphosphate. The release of inositol triphosphate or IP3 triggers release of Ca2 plus into the ovum cytoplasm. In response to the calcium signal, vesicles or granules present in the cytoplasm fuse with the ovum plasma membrane and release their contents. So we mentioned there must be some sort of release of inositol triphosphate or IP3, which triggers release of Ca2 plus into the ovum cytoplasm. In response to the calcium signal, vesicles or granules present in the cytoplasm fuse with the ovum plasma membrane and release their contents. The release of Ca2 plus in the ooze plasm provide the signal and the vesicles fuse with the membrane. These vesicles contain enzymes which cause a number of changes to occur in the ovum. Receptors are removed from both the ovum and sperm membranes. Receptors are removed from both the ovum and sperm membranes. Absorption of water molecules causes the ovum membrane to swell and push the vitelline membrane away from the plasma membrane. Absorption of water molecules causes the receptors are removed from both the ovum and sperm membranes. Absorption of water molecule causes the ovum membrane to swell and push the vitelline membrane away from the plasma membrane. 3. So vitelline membrane is pushed away from the plasma membrane. The vitelline membrane hardens and works as a block for entry of supernumerary spermatozoa. The vitelline membrane hardens and works as a block for entry of supernumerary spermatozoa. To understand and explain this process in detail, scientists recorded and videographed the process of fertilization in sea urchin and studied it in time-lapse photography. Time-lapse photography was conducted. The molecular changes were then described clearly. So directly beneath the sea urchin ovum plasma membrane are about 15,000 cortical granules, each about 1 μm in diameter. Upon sperm entry, these cortical granules fuse with the ovum plasma membrane and release their contents into the space between the plasma membrane and the fibrous mesh of vitelline envelope proteins. This process is termed cortical granule exocytosis, and several proteins are released in this process of exocytosis. The first cortical granule exocytosis, the first enzymes to be released are proteases. These enzymes dissolve the protein links that connect the vitelline membrane proteins to the cell membrane, and they remove the receptor molecule called the binding receptor. Sperms attach to this receptor during their binding process, and hence any spermatozoa attached to the binding receptor are also removed. Mucopolysaccharides released by the cortical granules produce an osmotic gradient. Mucopolysaccharides released by the cortical granules produce an osmotic gradient that cause water to enter into the space between the plasma membrane and the vitelline membrane, causing an envelope to expand and become the fertilization envelope. So then water is also going to enter, which is with the help of mucopolysaccharides, nothing else. In response, a third protein is released by the cortical granule. In response, a third protein is released by the cortical granule, a peroxidase enzyme, which hardens the fertilization envelope. A peroxidase enzyme which hardens the fertilization envelope. Peroxidase enzyme which hardens the fertilization envelope by cross-linking tyrosine residues on adjacent proteins. The peroxidase enzyme is there, which hardens the fertilization envelope. Fertilization envelope is also there. Peroxidase enzyme which hardens fertilization envelope by peroxidase enzyme. Peroxidase enzyme. Yeah. This fertilization envelope was created with the help of mucopolysaccharides. Yeah. So peroxidase enzyme which hardens the fertilization envelope by cross-linking tyrosine residues on adjacent proteins. As a result, the third protein was released by the cortical granules, a peroxidase enzyme which hardens the fertilization envelope by cross-linking tyrosine residues on adjacent proteins. As a result, the fertilization envelope starts to form at the site of sperm entry and continues its expansion around the ovum. As it forms, any superfluous bound sperm are released from the envelope. This process starts about 20 seconds after the first sperm attachment and is complete by the end of the first minutes of fertilization. In the last phase of this reaction, a fourth cortical granule protein, hyalin, is produced, which forms a coating around the ovum. The ovum extends elongated. Hyalin is produced, which forms a coating around the ovum. The ovum extends elongated microvilli, and the tips of these villi attached to this hyalin layer. This layer provides support for the blastomeres during cleavage. This layer provides support for the blastomeres during cleavage. Which layer? Microvilli layer. In sea urchins, the cortical granules secrete not only hyalin but a number of proteins that construct the extracellular matrix of the embryo. This is a highly coordinated process that results in sequential layers. In sea urchins, the cortical granules secrete not only hyalin but a number of proteins that construct the extracellular matrix of the embryo. The sequential changes ensure that the fertilization of ovum by additional sperm is prevented. They are part of the slow block to polyspermy. Though sexually reproducing organisms are adapted to avoid polyspermy, the defenses are particularly well characterized in the sea urchin, which respond to the acceptance of one sperm by inhibiting the successful penetration of the ovum by subsequent sperm. Similar defenses exist in other eukaryotes. Electrical polyspermy blocks therefore operate in many animal species, including frogs, clams, and marine worms, but this mechanism was not observed in several mammals that have been studied: hamster, rabbit, mouse. In species without an electrical block, polyspermy is usually prevented by secretion of materials that establish a mechanical barrier to polyspermy. Animals such as sea urchins have a two-step polyspermy prevention strategy with a fast but transient electrical block superseded after the first minute or so by a more slowly developing permanent mechanical block. It is, thought, it is true that electrical block evolved in those species where a rapid process for block to polysperm is needed due to the phenomenon of external fertilization by the presence of many sperm from varied species competing simultaneously at the ovum surface as occurs in animals such as sea urchins. In sea urchins, fertilization occurs externally in the ocean, such as hundreds of sperm can encounter the ovum within several seconds. Then, block to polyspermy in mammals. In mammals, fertilization occurs internally and fewer spermatozoa reach the site of fertilization in the oviduct. This is due to the fact that the female genital tract is adapted to minimize the number of sperm reaching the ovum. However, polyspermy averting mechanisms are essential in mammals to avoid the erroneous entry of several spermatozoa into the ooplasm. A series of biochemical changes occur in secretion reaction, the cortical reaction, which modifies the extracellular investments of the ovum, the zona pellucida. Further reaction results in stabilization and modification of the ovum's plasma membrane. The zona pellucida is modified by serine proteases that are released from the cortical granules. These proteases remove any receptors that may recognize and bind additional spermatozoa. Also, these enzymes digest the glycoprotein molecules between the cell membrane and the vitelline envelope. This helps to form the fertilization envelope from the cortical granules, which works as a barrier, blocking entry of superfluous spermatozoa. The cortical reaction marks the main event for block to polyspermy in mammals. Hence, species have evolved ways to prevent the union of more than two haploid nuclei in order to prevent the entry of more than one sperm into the ovum. In mammals, the cortical granule reaction does not create a fertilization envelope, but its ultimate effect is the same. So, released enzymes modify the zona pellucida sperm receptors such as they can no longer bind the sperm. During this process called the zona reaction, zona receptor molecules like ZP3 and ZP2 are modified. Scientists studying this phenomenon have proposed that the cortical granules of mouse ovums contain an enzyme that clips off the terminal sugar residues of ZP3, thereby releasing bound sperm from zona and preventing the attachment of other sperm. Cortical granules of mouse ova have been found to contain N-acetyl glucosaminidase enzymes capable of cleaving N-acetyl glucosamine from ZP3 carbohydrate genes. N-acetyl glucosamine is one of the carbohydrate groups that sperm can bind to, and when the N-acetyl glucosamine residues are removed at fertilization, ZP3 will no longer serve as a substrate for the binding of other sperm. So, N-acetyl glucosamine residues are removed. ZP2 is digested by cortical granule proteases and loses its ability to bind sperm as well. Thus, once sperm has entered and fertilized the ovum, other spermatozoa can no longer initiate or maintain their binding to the zona pellucida and are rapidly discarded. Role of calcium. Calcium plays a vital role in these proteases as the initiator of the cortical granule reaction. The mechanism of the cortical granule reaction is similar to that of the acrosomal reaction, which the sperm undergoes just before penetration through the zona pellucida. Upon fertilization, the intracellular calcium ion concentration of the ovum increases. If this calcium-rich environment, once again I'm going to read this because I'm kind of sort of confused. Calcium plays a vital role in this process as the initiator of cortical granule reaction. The mechanism of the cortical granule reaction is similar to that of the acrosomal reaction, which the sperm undergoes just before penetration through the zona pellucida. Upon fertilization, the intracellular calcium ion concentration of ovum increases. In this calcium-rich environment, the cortical granule membranes fuse with the ovum plasma membrane, releasing their contents. Once the fusion of the cortical granules begins near the sites of sperm entry, a wave of cortical granule exocytosis propagates around the cortex to the opposite side of the ovum. Once again, I'm going to read this and again confused. Calcium plays a vital role in those processes as the initiator of the cortical granule reaction. The mechanism of the cortical granule reaction is similar to that of the acrosomal reaction, which the sperm undergoes just before penetration through the zona pellucida. Upon fertilization, the intracellular calcium ion concentration of the ovum increases. In this calcium-rich environment, So calcium will be higher inside the ovum after fertilization. The cortical granule membranes fuse with the ovum plasma membrane, releasing their contents. Once the fusion of the cortical granules begins near the site of sperm entry, a wave of cortical granule exocytosis propagates around the cortex to the opposite side of the ovum. In sea urchins and mammals, the rise in calcium concentration responsible for the cortical granule reaction is not due to an influx of calcium into the ovum, but rather comes from within the ovum itself. The release of calcium from intracellular storage has been observed visually using calcium activated luminescent dyes such as aquarin or isolated from luminescent jellyfish. These dyes emit light when they bind free calcium ions. When the sea urchin ovum is injected with the dye and then fertilized, a striking wave of calcium release propagates across the ovum. Starting at the point of sperm entry, a band of light can be seen transverse the cell. Calcium ions do not merely diffuse across the ovum from the point of sperm entry. Rather, the release of calcium ions starts at one end of the cell and proceeds actively to the other end. The entire release of calcium ions is complete in 30 seconds in sea urchin ova, and the free calcium ions are taken up again shortly after they are released. If two sperm enter the ovum cytoplasm, calcium ion release can be seen starting at the two separate points of entry on the cell surface. Hence, nature has devised interesting and intricate mechanisms to ensure that only one spermatozoon fertilizes the ovum and that polyspermy does not occur, since it would result in a deformed embryo. Unlike mammals, this phenomenon has an interesting twist in birds. Birds' eggs are fertilized by more than one sperm, that is, by the process of polyspermy. Penetration by multiple sperm may be unusual in the animal kingdom, but it is somehow crucial to the development of the bird embryo. Experimental studies have verified this observation. Dr. Hemmings and her colleagues closely studied the developmental process in eggs from zebra finches and chickens. They inseminated the finches with fused sperm from 1 to 15 and compared these results with the naturally fertilized eggs. Under natural conditions, eggs in such birds may be fertilized by hundreds of sperm. Also, inseminated chickens with a little larger number of sperm. The separate studies, other chickens were inseminated with even larger numbers. So it's found that fused sperm penetrated the egg. The bird embryo was unlikely to survive. Survival has ensured that larger number of sperm fertilized the eggs. So that is something kind of weird in case of birds. Polyspermy in humans. In humans too, two main mechanisms operate to ensure that the block to polyspermy is complete. While the passage of ions and membrane charge change play a minor role, the main mechanism in the molecular changes during the cortical reactions and extrusion of cortical granules. The second proposed mechanism involves a Ca2 plus oscillation event activated by attachment of the sperm to the oocyte membrane. The subsequent release in intracellular Ca2 plus concentration triggers the exocytosis of cortical granules approximately 1 μm in diameter from just below the oocyte membrane into the periuterine space. Enzymes such as hydrolase, protease, and peroxidase, which are contained in the cortical granules, prevent the penetration of following sperm by modifying the structure of the sperm receptors such as ZP2 and ZP3. This results in the slow block to polyspermy by hardening the zona pellucida. The slow block occurs within approximately 5 to 8 minutes of oocyte activation and is considered the main mechanism of polyspermy block in humans. Historically, this process aroused the curiosity of several scientists who attempted to explain how among millions of spermatozoa only one is allowed to fertilize the ovum. Theodore Bovary's analysis of polyspermy is a classic experimental and descriptive biology which has outlined this process with interesting details. E. just studied these mechanisms extensively and provided delineation of the fast and slow blocks, and his findings were published in a critical paper in embryology. So now we are going to have a quick summary. The process of fertilization, gamete membranes fuse the sperm, nucleus, mitochondria, and centriole enter the womb, culminate in fertilization. Certain mechanisms are initiated to reduce the fusibility of the ovum membrane and block the entry of more than one sperm into the ovum. This is termed as blocked polyspermy. The cell resulting from polyspermy would have 3 or more copies of each chromosome, and the result would be a non-viable zygote. Two mechanisms exercise the blocked polyspermia as studied in the sea urchin. The first is known as the fast block and is initiated within 1/10 of a second of gamete membrane contact. Unfertilized sea urchin ova have a negative charge inside but changes to positive upon fertilization based on the exchange of sodium and potassium ions. Thus, after the first sperm contacts the ovum and causes the change, subsequent sperms are prevented from fusing. The slow block takes place 10 to 16 seconds after fertilization. Calcium is released and the fusion of the cortical granules begins at the site of sperm entry. A wave of cortical granule exocytosis causes water to enter into the space between the plasma membrane and the vitelline envelope, forming a barrier to any more sperm. While blocked polyspermia occurs in mammals and in some birds, however, polyspermia appears to be a natural phenomenon. So, thank you for listening.